Welcome to Radon's Smart Review RadBio edition.
And thank you for joining us for this comprehensive tutoring session today. We're diving into a topic that's really, central to modern radiation oncology.
Yeah, the incredibly dynamic and, well, increasingly important interaction between radiation therapy and the body's own immune system.
It's a huge shift in how we think about what radiation does, isn't it?
Absolutely. For so long, the focus was mainly just on, direct DNA damage, cell killing.
But now we see radiation also as like a powerful way to talk to the immune system.
Definitely. Our goal today is to unpack this connection to really understand the language radiation speaks to immune cells.
We'll cover how radiation affects things beyond the beam, explore the clever ways tumors try to hide from immunity.
Get to know the key immune cell players, who's helping, who's making things tricky.
Right. And then spend some real time on immune checkpoints like PD-1, PD-L1, and CTLA-4.
And critically, connect all that foundational biology back to the clinic. The exciting rationale for why we combine radiation with immune checkpoint inhibitors. This synergy is really transforming practice.
Exactly. By the end of this review, we want you to have a clear grasp of these complex interactions. Hopefully, you'll feel more confident, whether you're studying for boards or, managing patients on service.
Yeah, this is relevant stuff for residents at all levels.
Okay, let's start by thinking about how radiation's effects can actually extend beyond the precise area we're targeting.
We're talking about the abscopal and bystander effects.
These phenomena show us that hitting cells with radiation can trigger responses in cells that weren't even directly hit.
Right. The abscopal effect, that's pronounced abs-so-h-pol, is the one that really grabs attention.
Yeah, this is where local radiation to a tumor site leads to the regression of tumors outside that radiation field, sometimes quite far away.
And the key thing here, as you said, it's not due to physical dose scatter. This is fundamentally an immune-mediated effect.
The idea is that when radiation damages and kills tumor cells, it causes them to release specific signals, tumor antigens and inflammatory molecules.
Think of it like the dying tumor cells are putting out distress signals.
Exactly. And releasing bits of themselves as evidence.
And these signals then get picked up by the immune system, particularly those critical T cells we'll definitely talk more about later.
And if those T cells get activated against the tumor, they can then travel through the bloodstream to attack cancer cells at distant sites that never saw a single gray of radiation.
It's still pretty rare, right?
It is, yeah, historically rare, but it's much more frequently discussed and actually observed now in the context of immunotherapy.
Which makes sense because you're essentially hoping to unleash that systemic immune attack with the combo therapy.
Exactly. Now, a distinct phenomenon is the radiation-induced bystander effect. That's bystander. This is more about the local neighborhood.
Okay, so irradiated cells are signaling to their nearby unirradiated neighbors.
That's right. And those neighbors, they can show damage, things like DNA breaks or chromosomal aberrations, even though they weren't directly hit by the beam.
How do the signals travel?
Well, it can be through direct cell connections like gap junctions or through soluble factors released into the immediate environment around the cells.
So this is more confined, like right at the edge or near the irradiated volume.
Exactly. Unlike the abscopal effect, which is distant and systemic, bystander effects are pretty localized.
And is it clinically significant?
Well, that's still debated. Whether bystander effects contribute significantly to tumor cell kill just outside the field, or perhaps add to toxicity in nearby normal tissues. It's an ongoing area of research.
So both of these effects really highlight that radiation isn't just a simple physical event.
Not at all. There's complex communication happening between cells, and often the immune system is involved in that signaling chain. It just underscores the biological complexity.
And if the immune system is involved, that raises the question, why doesn't it just get rid of cancer on its own? Which leads us to tumor immune evasion.
Yeah, it's kind of frustrating, isn't it? Our immune system is built to spot and eliminate abnormal cells, including cancer.
But tumors have evolved these incredibly sophisticated ways to evade detection and fight back against immune attack.
Yeah, these are often called mechanisms of adaptive immune resistance. They really are clever survivors.
So what are some of the key strategies they use?
Okay, well, one major tactic is low immunogenicity. That's immunogenicity, N-I-S-E-T.
Low immunogenicity, meaning they don't look foreign enough.
Essentially, yeah. The tumor cells don't have enough easily recognizable flags or antigens on their surface for the immune system's T cells to spot them as dangerous. They just sort of blend in.
Like wearing camouflage. What else?
Another strategy is antigen loss or modulation. So even if the tumor starts with some recognizable antigens, the cells that happen to lose those antigens or lose their MHC molecules which are needed to display the antigens, those are the ones that survive immune pressure and grow.
So losing MHC makes them basically invisible to T cells.
Pretty much.
Hmm.
A true cloak of invisibility. Tumors can also engage in the production of immunosuppressive factors or cytokines, SY tokens.
They secrete molecules that dampen the immune response.
Exactly. They pump out things like TGF-beta, IL-10, PGE2. These tell anti-tumor immune cells to basically stand down. They create a hostile microenvironment.
And then there's the induction of immune tolerance. What's that?
So tumors don't just defend themselves. They can actively recruit and sort of educate certain immune cells to become their allies. They build this immunosuppressive ecosystem around themselves.
Which leads directly to a really key mechanism, one we absolutely need to understand for boards in clinic. Expression of immune checkpoint ligands, LYGANs.
Yes. Tumor cells, or other cells in the tumor neighborhood, can start expressing ligands like PD-L1.
And these bind to receptors on T cells and hit the brakes.
Exactly. It's a primary way tumors directly tell activated immune cells, stop attacking me.
Wow. Understanding these evasion mechanisms is just critical because it explains why targeting these specific pathways, like with checkpoint inhibitors, can be so effective.
Absolutely. And to really understand those checkpoints and how radiation interacts, we need to know who the players are on the immune battlefield.
Right, the tumor microenvironment. It's packed with different types of immune cells. Some are trying to help us and others, well, not so much.
Let's talk about the good guys. First, the anti-tumor immune cells.
Okay, who's top of the list?
That would be the CD8 positive cytotoxic SY-totokic T lymphocytes, lymphocytes, or CTLs.
The killer T cells.
Right. They are primary workhorses in adaptive immunity. They recognize specific tumor antigens presented on MHC class I on the tumor cells and directly induce cancer cell death.
The elite force, trained for specific targets. Who else is on our side?
Now we have natural killer, or NK cells. These are part of the innate immune system, kind of like the first responders.
Yeah.
They're particularly good at killing tumor cells that have lost MHC class I expression. Remember that evasion tactic we just talked about?
Yeah, so they counter that strategy.
Exactly. And NK cells don't need prior sensitization or training to recognize and kill certain stressed or abnormal cells. They're ready to go.
Good to have them. And then there are dendritic cells. Pronounced D-R-I-T-K.
Yes, dendritic cells, DCs. These are the professional antigen-presenting cells, APCs. Think of them as the intelligence gatherers and the drill sergeants for the T cells.
Okay, how do they do that?
They capture antigens from things like dying tumor cells, process them, and then they travel to the lymph nodes to present those antigens and activate the T cells, especially the CTLs.
So that antigen presentation step is absolutely fundamental for getting those killer T cells going.
Couldn't happen without them. Finally, we have macrophages, macrophages,
Right.
specifically the M1 phenotype.
M1. Are there others?
Yes, we'll get to M2 later. But M1 macrophages are classically activated. They can have anti-tumor functions, including directly killing tumor cells and releasing pro-inflammatory signals that help the overall anti-tumor response.
Okay, so those are the main good guys. CTLs, NK cells, DCs, and M1 macrophages. Now, who are the complicators?
Right. The cells that often end up helping the tumor or suppressing our anti-tumor immunity.
First.
Regulatory T cells, or Tregs. These are a specialized subset of T cells, but their main job is actually to suppress other immune cells.
So they dampen the activity of our killer CTLs.
Yes, and other fighters too. Tumors often recruit Tregs to create an immunosuppressive barrier. They use CTLA4 as part of their suppressive mechanism and produce inhibitory cytokines like TGF-beta and IL-10.
Like the tumor's personal bodyguard is telling our guys to back off. Okay, who's next?
Next are myeloid-derived suppressor cells, MDSCS. This is a pretty diverse group of immature immune cells that tend to accumulate in cancer patients, especially within tumors.
And they're suppressive too.
Very potent suppressors. They shut down both T cell and NK cell activity through multiple mechanisms like starving the T cells of nutrients or releasing reactive oxygen species. They just make it a miserable environment for our fighters.
Okay, Tregs and MDSCs. Anyone else?
And lastly, tumor-associated macrophages, TAMs. We mentioned M1 macrophages are good.
Right.
But TAMs, especially the M2 phenotype, are kind of educated by the tumor microenvironment.
And these M2 TAMs are bad news for us.
Unfortunately, yeah, often. These alternatively activated macrophages are frequently pro-tumor.
How so?
They can promote tumor cell growth, help build new blood vessels the tumor needs, that's angiogenesis, facilitate invasion and metastasis, and crucially, they suppress anti-tumor immune responses.
Wow. So understanding the balance between all these cell types within a tumor, like, are there more killer CTLs or are Tregs and MDSCs dominating? That's a major factor.
Absolutely.
Yeah.
It significantly impacts how aggressive a tumor is and how it might respond to different treatments, including radiation and immunotherapy. That balance is key.
Which leads us perfectly into our deep dive on immune checkpoints themselves.
Yeah, as we touched on, these checkpoints are like the natural safety mechanisms, the brakes, in our immune system.
They exist to prevent autoimmunity, right? Stop the immune system from attacking healthy tissue.
Exactly. But tumors are very, very good at exploiting these natural brakes on T-cell activity to turn off the anti-tumor response.
They hijack them. So the two main checkpoints we focus on clinically are CTLA4 and the PD-1, PD-L1 pathway.
Right. Let's break down CTLA4 first. That stands for cytotoxic T lymphocyte-associated protein 4.
Okay, CTLA4. Where is it found and where does it act?
CTLA4 is expressed on T cells, especially those regulatory T cells we mentioned, and also on activated effector T cells. Yeah. Its primary site of action, where it really applies the brakes, is early on.
Early on where?
During the T cell priming and activation phase. Yeah. And that happens largely in the lymph nodes.
Okay, so if you think of the lymph node as the T cell training academy.
That's a great analogy. When a dendritic cell, or APC, is presenting a tumor antigen to a naive T cell to train it, there are signals needed for that T cell to get activated.
Right, like a go signal.
A critical go signal comes from CD28 on the T cell binding to B7 ligands on the DC. But CTLA4 also binds to those same B7 ligands.
And it competes with CD28.
It does, and it actually binds more strongly with higher affinity. When CTLA4 binds B7, it sends an inhibitory signal into the T cell.
Ah, so instead of getting the go signal via CD28, it gets a stop or slow down signal through CTLA4.
Precisely. It's like CD28 is the gas pedal saying, go, activate. But CTLA4 is an early brake applied right there during the training session in the lymph node. It makes it harder for the T cell to get fully revved up.
That's a helpful way to think about it. It raises the threshold needed for the T cell to become fully activated and turn into a killer CTL.
Exactly. Okay, now onto the PD-1, PD-L1 pathway. PD-1 is programmed death protein 1.
And PD-L1 is programmed death lichen 1.
Right. Now, unlike CTLA4 acting early in the lymph node, this pathway primarily regulates T cell activity later in the process.
Later where?
Specifically in the peripheral tissues and right within the tumor microenvironment itself. PD-1 is expressed on activated T cells, including those CD8 positive CTLs that have made it into the tumor, as well as CD4 positive T cells, B cells, and NK cells.
Okay, and where do we find the ligand PD-L1?
PD-L1 is very frequently expressed by the tumor cells themselves, often upregulated as one of those evasion mechanisms we talked about. And it's also expressed by other immune cells within the tumor microenvironment. There's also a related ligand called PD-L2.
And when PD-1 on the T cell binds to PD-L1 on a tumor cell, what happens?
It delivers a powerful inhibitory signal to that T cell. Essentially tells it to stop.
What does that stop signal actually do to the T cell?
Well, this binding can reduce the T cell's ability to proliferate. It decreases production of important inflammatory cytokines like interferon gamma. It impairs its ability to kill cancer cells.
And it can lead to that state you mentioned earlier, anergy.
Yes, anergy, that's ANRG, which is basically unresponsiveness, or it can lead to T cell exhaustion. These are T cells that got into the fight but are now being actively shut down.
Okay, so if CTLA4 is the brake applied during training camp in the lymph node, the PD-L1 pathway is more like a brake applied directly at the battlefield site within the tumor, causing the fighting T cells to get tired and ineffective.
That's a really clear and important distinction to keep in mind. Tumors are smart. They upregulate PD-L1 precisely to use this brake and shut down the T cells that are trying to kill them, right where the action is.
This understanding of how tumors evade and how these checkpoints work brings us to, well, maybe the most exciting part of this whole intersection, radiation and immunotherapy, especially with immune checkpoint inhibitors, or ICIs.
Yeah, for a long time there was this idea that radiation in the immune system might even be antagonistic. High doses could be immunosuppressive.
But that thinking has completely flipped, hasn't it?
Totally flipped. We now see radiation as a powerful partner, especially when combined with these ICIs.
So what's the rationale? Why are these combinations so effective, at least in principle?
Okay, there are several key mechanisms, and they really build upon everything we've just discussed. First, and maybe most important, enhanced tumor antigen release and presentation. This primes the T cells.
Because radiation kills tumor cells.
Right. And when they die, they release all their contents, including tumor antigens. And crucially, they release neoantigens. That's NEOA antigens.
Neoantigens, those are the unique flags created by the tumor's specific mutations.
Exactly. And these antigens are then scooped up by our professional antigen-presenting cells, the dendritic cells.
And radiation helps the DCs too?
It can, yes. Radiation can also promote the maturation of these DCs and help them migrate to the lymph nodes. Once there, these educated DCs present the tumor antigens to T cells.
So you're essentially using the radiation to generate a personalized set of targets from the patient's own tumor,
Right.
and then presenting those targets to the immune system.
Precisely. The radiation is effectively turning the tumor into a personalized, in situ, meaning in-place vaccine that educates the immune system about what it needs to attack.
Okay, that's mechanism one. What's next?
Mechanism number 2. Radiation can induce a specific type of cell death called immunogenic, immunogenic, cell death, or ICD.
Immunogenic cell death, meaning the way the cells die actually stimulates an immune response.
Exactly. It's a particularly effective way for cells to die if you want to wake up the immune system, and that's where DAMPS come in.
DAMPS,
Yeah.
damage-associated molecular patterns.
Yes, DAMPS, often just called DAMPs. These are molecules normally found inside the cell that get released or become exposed on the cell surface when a cell dies in this immunogenic way.
And they act as danger signals.
That's the perfect term. They act as danger signals to the innate immune system, alerting it that something bad is happening and help is needed.
Okay, so what are some key DAMPS released by irradiated cells that we should know?
Let's highlight 3 important ones. First, calreticulin, K-A-L-reticulin, or CRT.
Calreticulin, what does it do?
Normally it's inside the cell, but in ICD, it gets exposed on the surface of the dying tumor cell. It acts as an eat me signal, specifically targeting dendritic cells and promoting their phagocytosis, Phagocytosis.
Phagocytosis means engulfing the cell.
Exactly. It tells the DCs to engulf the dying tumor cells, which is crucial for picking up those antigens we talked about.
So CRT is literally telling the DCs, here, eat this, it's important.
Essentially, yes.
Uh-huh.
Second, DAMPHMGB1. This protein is released from the nucleus of dying cells.
Okay, HMGB1.
It binds to receptors like TLR4 on dendritic cells. This binding helps the DCs mature and produce interleukin-12, which is a cytokine really important for developing strong T helper 1 and CTL responses.
So HMGB1 helps the DCs get ready for serious business and train the right kind of T cells. Got it.
Third, DAMP.
Third one is ATP, adenosine triphosphate, the cell's energy currency.
ATP.
Yeah. When released from dying cells, ATP binds to specific receptors on DCs and helps activate something called the inflammasome.
The inflammasome, what does that lead to?
Activation of the inflammasome promotes the release of another cytokine, interleukin-1 beta, and IL-1 beta also aids in priming those T-cell responses.
So ICD and these DAMPS, calreticulin, HMGB1, ATP, they're critical signals.
Yes.
They ensure the DCs don't just clean up debris, but get properly activated to launch a real anti-tumor immune response.
Exactly right. They translate cell death into immune activation. Okay, mechanism number 3 for synergy. Modulation of the tumor microenvironment. We called it the battlefield earlier. Radiation changes the conditions on that battlefield.
It really does. Radiation can help convert an immunologically cold tumor, one that has few infiltrating T cells and is generally immunosuppressive, into an immunologically hot tumor.
A hot tumor meaning more welcoming to immune cells.
Yeah, more immunopermissive,
Yeah.
immunopure, and importantly, infiltrated by effector T cells like those killer CD8 positive CTLs.
How does radiation make it hotter?
Well, it can increase the infiltration of T cells and other helpful immune cells. It can cause the tumor cells to upregulate MHC class 1 expression, making them more visible targets for CTLs again.
Okay.
And it increases the production of pro-inflammatory cytokines and also chemokines. Those are molecules that act like signals to attract immune cells to the tumor site.
But wait, you mentioned earlier that tumors can still evade even if T cells get in. And didn't you also say radiation can sometimes even upregulate things like PD-L1 on the tumor?
Excellent point. And that is precisely where the combination really shines. That brings us to mechanism number 4, overcoming T-cell exhaustion or resistance.
Oh.
So even if radiation primes T cells and they make it into that now hotter tumor microenvironment, the tumor can still apply that PD-1 brake we talked about.
Right, causing those infiltrating T cells to become exhausted and ineffective.
Exactly.
Yeah.
And this is where the immune checkpoint inhibitors come in, right? To release those brakes.
That's their job here. That's their core function in this combination context. Drugs that block CTLA4 or the PD-1, PD-L1 interaction prevent the tumor from shutting down the immune response that radiation is trying so hard to generate and attract.
So the different types of inhibitors work at different points.
Generally, yes. Anti-CTLA4 tends to act earlier, helping prime that initial response back in the lymph node training camp. Anti-PD-1 or anti-PD-L1 acts later, right in the tumor microenvironment, helping to reinvigorate T cells that are getting shut down right there on the battlefield.
And by unleashing this radiation-primed, locally inflamed immune response and potentially making it systemic.
You increase the chances of seeing that rare but really exciting abscopal effect, which we can call mechanism number 5.
Exactly. It's the combination radiation acting as the in situ vaccine and microenvironment conditioner, plus the IC is releasing the brakes on the resulting immune response that makes the abscopal effect more likely to be observed clinically.
This area is evolving so fast. What are the key immune checkpoint inhibitor drugs that residents really need to be familiar with right now?
It is an expanding list for sure, but let's name the major players you'll definitely see and hear about. For the CTLA4 inhibitors, the landmark one is ipilimumab. That's IPII-limumab, trade name Yervoy.
For PD-1 for CTLA4, what about PD-1 inhibitors?
For PD-1 inhibitors, the most common are nivolumab, nivolumab, which is Opdivo, and pembrolizumab, pembrolizumab, Keytruda. There's also cemiplimab, say MIP-limab, trade name Libtayo.
Nivolumab, pembrolizumab, cemiplimab for PD-1, and PD-L1 inhibitors.
For PD-L1 inhibitors, atezolizumab, ATEG-azolizumab, Tecentriq, durvalumab, Imfinzi, and avelumab, adviolumab, Viventia.
Atezolizumab, durvalumab, avelumab. Okay. And are there newer targets emerging too?
Yes, definitely. For example, LAG3 is another checkpoint. There's a drug called relatlimab, RELATLAM, that targets LAG3, and it's actually given in combination with nivolumab under the name Opdualag.
These combinations have really changed practice, haven't they? The Pacific trial is probably the example most residents know well.
Absolutely. Pacific showed the clear benefit of adding durvalumab after chemoradiation for stage 3 non-small cell lung cancer.
Right.
It's a landmark study showing this synergy really translates into improved patient outcomes, establishing a standard of care.
Okay, fantastic. Let's try to pull together some clinical pearls, those absolutely need-to-know points for rotations and definitely for board exams.
Sounds good. Pearl number 1. The abscopal effect is regression of distant, non-irradiated tumors after local radiation, and remember, it is immune-mediated.
Pearl 2. Know your cellular army. CD8 positive CTLs are the direct killers. NK cells are innate killers, especially for cells missing MHC1. Dendritic cells are the crucial APCs that prime T cells.
And on the other side, regulatory T cells, Tregs, are suppressors. Myeloid-derived suppressor cells, MDSCs, are also suppressors, and M2 tumor-associated macrophages, TAMs, are generally pro-tumor and suppressive.
Pearl 3. Immune checkpoints act in different places primarily. The PD-1, PD-L1 pathway acts mainly in the tumor microenvironment, leading to T-cell exhaustion. CTLA4 acts earlier in the lymph nodes during T-cell priming.
Pearl 4. The core rationale for combining radiation plus ICIs. Remember, RAD acts as an in situ vaccine, antigen release, DC maturation, and conditions the microenvironment, makes it hotter via ICD and releasing DAMPS like calreticulin, HMGB1 ATP. Then the ICIs release the brakes, PD-1, PD-L1, CTLA4, on the resulting immune response.
Pearl 5. Associate the key ICIs with their targets. CTLA4, ipilimumab, PD-1, nivolumab, pembrolizumab, PD-L1. Idualimino, atezolizumab, durvalumab. Memorize those pairings.
Okay, moving just to beyond the foundational stuff, let's briefly touch on some ongoing research areas and, clinical decision-making challenges.
Yeah, because this field is definitely still rapidly evolving. There are big questions we're still figuring out.
For sure. One major area of investigation is the optimal sequencing and timing. Should radiation come before, during, or maybe even after the ICI to get the best immune priming and synergy?
What's the thinking currently?
Well, a lot of evidence so far often points towards giving radiation either before or concurrently with the ICI. The idea is you get better priming that way.
It makes intuitive sense, right? Generate the targets and the inflammatory signals with radiation, then release the brakes on the immune system with ICIs.
Exactly. Another crucial area is finding reliable biomarkers. How do we predict which patients are most likely to benefit from these combinations? It's not everybody.
What are people looking at?
Researchers are studying things like PDL1 expression levels in the tumor, the total tumor mutational burden, TMB, or specific gene signatures related to immune activity, like interferon gamma pathways.
But it's complicated.
It's not simple, yeah. No single biomarker is perfect yet, but we're definitely learning more about how to select patients better.
And clinically, managing the toxicities is huge, right?
Absolutely paramount. Combining therapies can lead to this complex mix of radiation-related side effects and then these unique immune-related adverse events, the IRAEs.
And those immune toxicities can affect almost any organ system.
They can. Recognizing and managing them effectively, often working closely with our medical oncology colleagues, is a really critical skill in this era of combined therapy.
Definitely requires vigilance and teamwork. Okay, ready for a quick board review blitz to test some of these concepts.
All right, let's do it. Hit me.
Question 1. A patient receiving radiotherapy for a primary lung tumor shows regression of metastatic liver lesions that were not in the radiation field. This phenomenon is known as A. Bystander effect, B. Tumor lysis syndrome, C. Abscopal effect, D. Reoxygenation. Keywords: distant, non-irradiated regression.
Okay, that's got to be C. Abscopal effect. Distant immune-mediated regression. Classic definition.
Correct. Question 2. Which of the following is a key effect? Damage-associated molecular pattern, or DAMP, exposed on the surface of irradiated tumor cells that promotes their phagocytosis by dendritic cells, contributing to immunogenic cell death. A. PD-L1, B. TGF-beta, C. Calreticulin, D. interferon gamma. Think eat me signal for DCs.
That's C. Calreticulin. It moves to the surface, signals phagocytosis, helps the DCs grab those antigens.
Exactly. Question 3. The PD-1 receptor is primarily expressed on which cells? And its engagement with PD-L1 typically leads to what outcome? A. Tumor cells, leading to tumor cell apoptosis. B. Dendritic cells, leading to enhanced antigen presentation. C. Regulatory T cells, leading to immune suppression. D. Activated T cells, leading to T cell exhaustion or inhibition. Where does PD-1 act?
Okay, PD-1 is on the T cells, the activated ones in the tumor. Binding PD-L1 shuts them down, so that's deactivated T cells, leading to T cell exhaustion or inhibition.
Perfect. Last one. Question 4. The primary site of action for CTLA-4 in regulating T cell responses is A. Directly on tumor cells, inducing apoptosis. B. Within the tumor microenvironment, blocking T cell effector functions. C. In lymph nodes during the initial priming and activation of T cells by antigen-presenting cells. D. On endothelial cells, preventing T cell extravasation into the tumor. Think back to that training camp analogy.
Right. CTLA-4 is the early break during priming. That happens in the lymph node, so it's C. In lymph nodes during initial priming by APCs.
Excellent job on those. Covers some really core points.
It does. So to maybe wrap things up and summarize this session, we've covered the really complex and, frankly, exciting interplay between radiation therapy and the immune system.
Yeah, we talked about how radiation can have effects beyond the abscopal and bystander effects.
We explored how tumors aren't just passive targets, but actively use strategies like low immunogenicity and checkpoint ligand expression to hide from or fight off the immune system.
And we identified the key cellular players on that tumor microenvironment battlefield, the good guys like CTLs, NK cells, DCs, and the complicating factors like Tregs, MDSCs, and M2 TAMs.
Crucially, we broke down the two major immune checkpoint pathways. CTLA-4, acting early in lymph nodes during priming,
Right.
and the PD-L1 pathway, acting later in the tumor microenvironment to cause T cell exhaustion.
And we put it all together to really understand the powerful rationale for combining radiation with immune checkpoint inhibitors, how radiation acts as that in situ vaccine,
Generating antigens, maturing DCs.
and conditioning the microenvironment through ICD and DAMPS like calreticulin, HMGB1, ATP.
While the ICI is then release the brakes, blocking CTLA-4 or PD-1, PD-L1, allowing those radiation-primed T cells to actually do their job effectively.
And this synergy is exactly why combinations using drugs like ipilimumab for CTLA-4 or nivolumab, pembrolizumab, durvalumab for the PD-1 axis are becoming standard practice and represent such a major advance.
Understanding these principles is just fundamental now for residents navigating this field. It's certainly a lot of biology to keep straight, especially when you're thinking about board exams.
It really is. You know, sometimes remembering all these immune pathways, cell types, and checkpoint inhibitors after your boards feels a bit like trying to find a parking spot on the first day of residency, almost impossible.
Huh. That's painfully accurate. Hopefully this review helps make that daunting task just a little less impossible.
We sincerely hope so. Complete practice oral boards at radoncssmartlearn.com, pronounced radoncssmartlearn.com, and subscribe to Radonc Smart Review for our next episode.
Thanks for tuning in.